Welcome to Fuzzy Butts and Friends. Well, as much as it drives Fuzzy Butts all over this place absolutely crazy when they hear me say it, I am the host of this show, your big dog, and I'm back again with you this week. And look over my left and right, and that's not Ginger Morgan, our co-host and co-producer. She will not be with us this week. Unfortunately, she is doing a media event, which is important and great, but it's not a media event for Fuzzy Butts and Friends, unfortunately. So I need to talk to my co-producer about that. But I'm glad, happy to have one of this, what I think is a really great figures in the world of veterinary oncology with us today. So since Ginger's not here, we get to talk all about data, not anecdotal funny things. And it's Dr. Doug Thamm. He's with the Flint Animal Cancer Center at Colorado State University. Dr. Thamm, welcome to Fuzzy Butts and Friends.
Oh, thanks very much for having me.
Great. Well, we like to start the show off with a little bit of levity and a conversation about fuzzy butts, since this show is called Fuzzy Butts and Friends. So do you currently have any fuzzy butts yourself?
I sure do. I have a 6.5-year-old female spayed mini golden doodle. I feel like a little bit of a chump being a guy with a designer dog, right? But my justification for that is my wife has pretty severe pet hair allergies. So we wanted to go for one of those breeds that had a pretty high likelihood of being hypoallergenic, and it worked out great. So our particular dog, has the right genes, so she really is hypoallergenic. So works out great for our family.
What's the name of your golden doodle?
Her name is Bindi, Bindi.
Bindi. That's an unusual name. I would say it was inspired by Steve Irwin's daughter, but I think her name is spelled Bindi. What's the origin of the name Bindi, if you don't mind?
So my dog's name is spelled exactly the same, Bindi,
Oh, I
but it
missed
was
that.
not named
Sorry.
after. Yep, but it was not named after Steve Irwin's daughter, actually. So the little dot that Indian women have on their forehead is called a bindi, although it's spelled with an H in there. And when my dog was just a tiny, tiny puppy, she had a little white spot right here on her forehead. So that's where the name came from. But interestingly, she sort of outgrew the little white spot as she reached adulthood. So she still got the name, but she doesn't have the spot on her forehead anymore.
All right. Well, it's a great name for a golden doodle. All right. Why don't we start next then with your origin story, sort of how you began your journey, your pathway into veterinary medicine, and where you're at now. You've done so much work. I was on reading your CV this morning, and you've published so much. You've run so many clinical trials, and you have so many accolades, and you're involved in so many different things. I had Craig Clifford on recently, and I think he's been on our show 3 times now, and he's a good friend of mine. He's like, "Have you had Doug yet on Fuzzy Butts and Friends?" I'm like, "No, I don't know why." So tell us how you started with the love of animals, and then got to where you're at with all the wonderful things that you're working on.
Yeah, so I mean, I think the origin story is pretty typical for a veterinarian. I grew up being like one of those animal kids. So, had a dog, had a cat, had I think the maximum was 7 tanks of fish that my parents would let me have in the house. And I'd always be the guy out there going out and catching box turtles and snakes and stuff like that. Did that through most of my childhood. Was kind of a horsey kid growing up also. So rode horses, and it just seemed like the large majority of the stuff I was interested in at the time was either related to animals or related to music. And got some advice along the way saying music is a great pastime. It's kind of a crappy way to make a living. So gravitated toward the animal thing instead. And that was really again, with the exception of this detour, hey, do I want to be a professional musician instead? I pretty much was focused on the idea of doing stuff that was related to animals and specifically veterinary medicine, probably from junior high all the way up through college and right into vet school. So yeah, typical story you'd probably hear from a lot of veterinarians as far as that goes. The oncology thing. So in undergrad, I had a chance to actually work at the veterinary school in a lab that wasn't having anything to do with oncology. It was actually a microbiology lab, but I really kind of got the research bug at the time, and I said, I think I still want to be a veterinarian, but I think I want to have a career that has something to do with research. Like I don't think I'd be happy or fulfilled just kind of hanging out a shingle and doing general practice. So God bless those people. That's an awesome thing, but I just didn't think that was what was going to keep me going through the years. So kind of said, I think I want to do something that involves research, but didn't really know what that was. And had a personal cancer experience actually coming up through vet school. Took a year off actually being treated for non-Hodgkin's lymphoma. And it is not one of those things that some people say, oh, I had this experience and it changed my life, and I was going to dedicate the rest of my life to stomping out cancer. Like it really was not an experience like that. So I went into it with a very, very, very favorable prognosis. So everybody kind of said, this is going to be a really inconvenient year, and it's going to be a drag, but you're going to be fine. And but the one thing it did do was kind of made me just think about oncology in animals. So this is back in the 90s. The number of like card carrying oncologists that were out there, you could probably count on two hands back then. So it wasn't anything like it is now. But I did say, geez, I wonder what that's like in animals. Like I hadn't really encountered it in the first year of my veterinary curriculum. And yeah, I found out that it was a thing. And at the time, I got my veterinary degree at University of Pennsylvania, and there was a gap in sort of oncology service there. So Ann Jeglum had left at that time, and the oncologist that they hired, Karen Serenmo, hadn't quite started. So when I was there was no oncology. So I actually had to go far afield and actually make a trip to Wisconsin in order to experience clinical oncology as a fourth year student. Had an awesome experience there with Greg McEwen and David Vale, and that basically nailed it for me. I said, "Yep, this is great. I think I want to go do specialty training in oncology, and hey, wouldn't it be great if they would take me at University of Wisconsin?" So that's kind of what happened. So ended up at Wisconsin for what was originally a 2-year residency. So I was one of the last of the 2-year residents back when that was feasible. But I lobbied to have that turned into a 3-year program because I wanted more opportunity to do research. And they managed to scrape up the money to turn that into a 3-year program. And I actually ended up staying for 2 more years doing a postdoc there after my residency was complete. Then took a year and actually spent a year working at the University of Queensland in Australia for a guy called Rod Straw, who is actually a pretty well-known surgical oncologist, trained here at CSU back in the day. So spent a year there. Amazing, amazing, amazing country, amazing year. I probably never would have come back if it wasn't for the huge amount of student loans that I had to pay. And the Aussie dollar was way too weak for me to pay those loans back at the time. Ended up sort of having a hybrid position, so working 3 days a week in a private practice in Milwaukee, working 2 days a week in the lab still at Madison for 2 years. That takes us to 2004, which is when I applied for and got the position here. So this November is going to be my 20-year anniversary here at CSU.
Cheers to
And
that. Well,
that's
you
been
met
my origin story.
Cheers. That's quite an origin story, and it
Yeah, yeah.
includes Australians too as well. But so McEwen was a part of directing you early on, and as I know, he's one of the grandfathers of veterinary oncology, along with Withrow. So you both were interacted with McEwen and Withrow there at CSU. So did you work a lot under?
Oh, I'll do you one better than that. So yeah, so I mean, I had a chance to work with one of the fathers of medical oncology, one of the fathers of surgical oncology, but I also got a chance to overlap for several years with Ed Gillette here, who was one of the fathers of radiation oncology. So I really got to learn from the best in all three of those disciplines, which was a pretty special opportunity.
Yeah, it most certainly is. What is cancer like? You did a year, I think you said a year in Australia. What is the prevalence of cancer over there? What does it look like?
Oh, that's a great question. So, I mean, a lot of the cancers that we see are the same. We see our lymphomas and our mast cell tumors and our osteosarcomas. Uh, Australia is such a sunny place that we do see a lot of those sun-induced skin cancers that we see here too in both dogs and cats. So there was a little bit of an overabundance of that. And one of the interesting things is that they have a really weird fungus in Australia that can look a lot like cancer. So we see that sometimes here in the United States if you practice far down south or way up north. There's some funguses that can appear in the bones and in the lungs that can look like cancer. And they have that in Australia too, but it's actually a different fungus. So it's a fungus called aspergillus that can look a lot like bone tumor, especially in German Shepherds, which is a really interesting thing. So whatever the reason is, German Shepherds get a lot of this. A lot of the other breeds not so much, but that was one of those confounders, right? One of those cancer mimics that we had to worry a lot about there. But yeah, other than that, it was the skin cancer that we really saw a lot of, and that's definitely the case in people too. That's like skin cancer capital for humans as well.
Well, 2004 was also a pivotal moment and year in my lifetime. It was the year my first dog, Malcolm, was diagnosed with bone cancer. I was up in Waltham or Watertown, Massachusetts, and he started limping, and I took him to see a vet in Waltham. And he took a radiograph of it, and ironically enough, he took one look at it, and I'd never seen the starburst pattern before. I now know what that looks like, and I'm always scared of it. But he said, yep, 99% certain it's cancer, 1% chance it's fungus. So he gave it a 1% chance that I guess it was the fungus that we sometimes confuse with cancer. And of course, it turned out to be osteosarcoma. And that changed the course of my life. It went on to spread to his lungs. I moved back to Texas. And I'm not sure if you're familiar, but I started my first walk across the country from Austin to Boston and formed Puppy Up. So it's 20 years for me. I've been dealing with cancer and companion animals, so I'm not a novice. And I've since lost two more dogs to cancer. One to nasal adenocarcinoma, which he was treated at CSU by the radiologist there, Susan, and as well, Dr. Withrow, of course, helped out because I know him or knew him fairly well back then. And so there's a for me, I love CSU. There's a big CSU connection in my history, and they took such great care of Murphy. But okay, so we got a lot to cover today, and you can talk about these so these bullet points as much. You can elaborate as much as you want, or as little as you want. Take it any direction. But I kind of want to take a snapshot of your 20 years now at CSU and talk just kind of broadly a little bit about cancer and companion animals. What have you learned? What are you seeing? What are some things you would like our audience to think about?
I mean, I think in very, very general terms, one of the things that we spend an awful lot of time doing here when we're talking to pet owners is really sort of dispelling some of the myths about cancer treatment in animals that may make some owners kind of not even come to see us in the first place. They may get a cancer diagnosis from their regular vet and they say things like, "Oh my gosh, I would never do surgery for cancer on my pet. I would never do radiation. I would never do chemotherapy." And I think, the audience of this podcast probably is pretty well informed. But I think one of the great things that your listeners can certainly do is spread that word that, hey, cancer treatment in animals, yeah, we use the same tools. We use radiation therapy, we use surgery, and we use chemotherapy, but the way that we do it is very, very different. So a lot of the side effects that people associate with cancer treatment in people are just really not there to the same degree in our pets. And we're much, much, much more interested in focusing on quality of life and making sure they can keep doing all the regular stuff that they've been doing their whole lives and enjoying themselves and all those kinds of things. And I think making sure that everybody out there sort of understands that component of it is, gonna make the discussions that we have with owners that much easier because they already come in with that information. But B, I think it's gonna make it so that more owners at least go in to talk to a veterinary oncologist about their options if they're not scared away by the prospect of all these side effects that they're kind of thinking about based on personal experience or family experience or experience with their friends. So if I was going to sort of say deliver one message to kind of the public out there, it's going to be that it's like it's not necessarily like what you're thinking, and come in and talk to us before you make a decision about what you want to do. I think that's the biggest take home I think I could give.
It is. It is. That's the most important thing. It's mostly treatable, and you can have some positive outcomes. And the thing I say almost in every episode that we talk to a veterinary oncologist is my advice to pet parents is go see a board certified veterinary oncologist. As you know, the general practitioners comes in all flavors and sizes and forms, and some of them are not up to date or current with all the different treatment modalities we have and all the new latest in therapeutics and diagnostics that are coming out. And that's my first advice is that if you suspect your companion animal has cancer, go straight to see a board certified veterinary oncologist to get the right data to really find out what all the options are and potentially what the outcomes may be.
Yeah, I mean, the other thing that I'd add to that is, by committing to go in and talk to a specialist, you're not committing to doing anything, right? You're just information gathering. So, you're not saying I'm gonna treat this way or I'm gonna treat that way or whatever. It's just getting options. And I think that can be really liberating and informative for an owner, even if at the end of the day the owner said, yeah, I decided maybe I'm not gonna treat super aggressively, but I'm really glad I got the information. So I think that's another sort of important take home. It's just information gathering if you come in and talk to us.
Absolutely. Well, as you know, that even though the supply pool of veterinary oncologists has expanded considerably since 2004, it's still way too small. And I knit about
Yeah.
that with Phil Bergman and Craig Clifford all the time. Is what could pet parents do to get to expand the pool of board certified veterinary oncologists? Because I'm sure, as you know, some states don't have them, and it's a bit of a burden. But
Yeah.
like you were saying, is my advice to them is always it's worth the trip. It's however many hours you have to drive, maybe stay overnight in a hotel. That data can change the outcome of your companion. You could have a very treatable and form of cancer, and you could get a very good outcome and have several years of life. And as you know, several years to a dog that only lasts 12 years is 25-30% more life, and everybody wants that. So that's good information. Well, let's talk. You mentioned the three forms of cancer treatment. I find it interesting that you left out immunotherapy because I was looking at your CV and you recently published some research on immunotherapy. So that's becoming a treatment option too now for our canines with cancer, right?
Well, we're really just scratching the surface in veterinary oncology about sort of knowing how to utilize those tools. And, we're probably conservatively 20 years behind where they are on the human side. So, I mean, it's a super exciting time, right? So we do have some really interesting new tools in our toolbox, like this immune checkpoint antibody that we actually can't currently get, but is kind of out there. But again, we're just scratching the surface about how to use that. What are the best tumor types to use it for? Is there some way we can pick out who it's going to work for and who it's not going to work for ahead of time? I mean, a million questions there that that we're still looking to answer. Um, there are some other antibodies out there that may be coming up in the near future. So lymphoma targeting antibodies, for example, that they've had for humans for 20, 30 years, that we may see come around pretty soon that have the potential to really significantly improve the outcome there. So, boy, it's a really, really exciting time. But I got to be perfectly honest, we don't quite know what we're doing yet with with most of the immunotherapy that's out there. So it's a work in progress.
Well,
Stay tuned.
that's good to know. But it's interesting that I've always said, and I've spoken to so many oncologists on my travels, which have been very long and many, and I've always said that 20-year gap between human medicine and veterinary medicine, it's legit. Do you ever see, Dr. Thamm, do you ever see that gap narrowing? Do you ever see, I know it's a function of money, resource allocation, and a number of other things, but I think everybody realizes now, I think the number we're using is 6 million new cases of cancer in dogs every year alone. I mean, there's money there for big pharma. There's money now there to be made. And I think that the pharmaceutical industry is now getting up to snuff on that. Do you ever see that? Do you ever see veterinary medicine accelerating a little bit faster?
Well, I mean, I think, yeah, I think I do. And, some of those have to do with practical things. So, one of the big sort of gaps in our knowledge for a long time has been, what are sort of the molecular underpinnings of the dog tumors that we see, right? So what's wrong with dog DNA or dog tumor DNA that makes these things happen, and how is it the same or different from human DNA? And as the tools get cheaper and cheaper to be able to answer those kinds of questions, yeah, we're actually learning a lot more about that. So I think our sort of fundamental knowledge base is actually catching up a little bit just because those technologies are getting cheaper and more ubiquitous. And I mean, I think if you take a look at sort of where things have been going in the last few years, there are a lot more new treatments that are sort of dog specific that are out there. If we were looking 10, 12 years ago, we had Palladium. That was it. That was the dog drug. And we had the Oncept melanoma vaccine. So now you can add Tenovia, you can add Laverdia, you can add crofelemer, you can add the the thing that you shoot into the mast cell tumors, whatever the name of that is. It's escaping me at the moment. That's kind of embarrassing. Stelfonta. We've got the anti-PD-1 monoclonal antibody. So I mean, I do think that we're starting to see a little bit of a kind of a rev up in the sort of dog specific treatment options that are out there. So on both fronts, I think, yeah, I mean, I'm not sure we're ever going to catch up to the human side, unfortunately, but I think the tools in our toolbox are getting a little bit more in number, which is exciting.
Yeah, I always refer to it as that tools. That's exactly what pet parents want. We want more tools in our growing toolbox, and also our general understanding of cancer. But as you know, that changes as well. Cancer is not a static thing. It's not a stationary target, and unfortunately. Um, well, let's talk about what I guess before I forget it, I made a note of it. It wasn't on the bullet points, but one thing I'm a big fan of and I like to research in my spare time is artificial intelligence. Do you is CSU are you guys doing any research with AI right now as a diagnostic tool at all?
Um, so funny that you mentioned that. So we have some very, very, very early studies that are going on, trying to start to use some AI predictions for outcome assessments. So one of our doctors here is very, very interested in using AI to see if we can predict outcome in cats with high-grade lymphoma. So that's kind of an interesting one. About half of those cats can do real well. The other half really don't. We don't know how to pick out one versus the other. So she's actually very keen to look at sort of AI-informed clinical things. So blood work results and signalment information and, all those kinds of things to try and see if we can better predict who's going to do well and who's not. And she wants to apply some of those same tools actually to prediction of adverse effects from chemo in dogs with lymphoma. So like we are literally just getting started with those. So, we're not going to have anything to tell you for a couple years probably on that front. But yeah, so we are starting to make some inroads there. We also have some pathologists who are actually certainly interested in using AI-derived tools for, looking at new markers under the microscope and things like that. There's some neat tools that are available for helping the pathologists do that stuff too. So, but again, that's definitely like stay tuned for for future news flashes about that. We are just scratching the surface
Yeah,
here at
well,
CSU on that.
right. Well, as you know, AI is still such a nascent technology. I mean, broadly a nascent technology for all industries and all use cases, it seems like. So it's going to take a couple of years, but I'm excited to learn that there are researchers there at CSU that are interested in it because it will be such a big part of the future. We know that. We've had Dr. Jaime Modiano on the show a couple of times, and he's doing the same up there in Minnesota. So I'm glad to hear CSU is working on that. Well, I'm glad you had an answer. I'm glad I asked that because I didn't want to forget about that. And I'm an AI nerd, and I'm excited about all of its potential. And it will be a tool. That will be a very important tool in our toolbox. Okay, one thing I wanted to ask you, Dr. Thamm, is what are your areas of interest? So you're a researcher. You've published a lot of papers, but broadly, what do you like to work on? What areas of cancer are you interested in?
I'm for better or for worse, and some people sort of consider this one of my faults. I'm pretty agnostic when it comes to what kind of cancer I study. So I think it's much more about, is there an interesting question to be answered? Is there a new treatment that somebody is interested in having me help out with? Um, and there are different tumor types that might work best for different specific treatments. So I think it's much more the question that's being asked rather than the individual tumor type that kind of gets me excited. And that's true both for kind of the clinical research that we do here, but also the work that I do in the lab. So it's some people, like sort of potential grad students, actually sometimes find that very frustrating. So, they come in, they sit down with me and they say, "Hey, what do you work on?" I might be interested in working in your lab for my PhD, and I'll tell them, this time next year, I can't tell you what my lab's going to be working on because it's broadly going to be something having to do with animal cancer and maybe something to do with, using animal cancer as a model of the human disease, but I can't tell you what kind of tumor type it's going to be. I can't tell you what drug it's going to be because again, that's just something that is really much more about the question than it is the treatment modality or the tumor type. I think if you kind of look at kind of the work that we've done over the years and just sort of count up papers, I think probably the one that comes to the top of the list is lymphoma for me. But I think that has more to do with what walks through the clinic door, more so than it's because it's a unique passion of mine. It's just, yeah, you very much use what's out there, use the clinical material that you're seeing, and try to answer questions. And that happens to be what we see a lot of, just like all the other medical oncologists that are out there. So I think you're gonna see a lot of papers with my name on it that have to do with lymphoma. You're gonna see a lot of papers with my name on it that have to do with mast cell tumor. Same thing. We see a lot of mast cell tumors. So it's got more to do with that, just because that's what we see, more so than that's a unique or special passion of mine. It's just there's lots of questions to be answered with those tumor types, and we see enough of them that it's maybe a little easier to answer some of those questions in those tumor types than it is for the more rare stuff where we just don't see a lot of it, and it's hard to answer those questions. So yeah, so I'm a jack of all trades. I'll study any old kind of canine cancer if the question's right.
I dig that. I've always sort of thought of myself like that as well. But what sort of interesting questions? Let me narrow the scope of it a little bit. What sort of interesting questions to be answered are perturbing you right now? What are you looking at? What are you interested in right now?
Um, so we have a bunch of different things that are going on. I think there's two that I'd point out that I think are kind of maybe uh, a little more practical, or might have a little bit more immediate impact than some of the other stuff that's going on. And one of these is a study that we've got funded by the American Kennel Club, actually, that's looking at a new treatment protocol for dogs with lymphoma. And the thing that's unique about this particular treatment protocol is we designed it to only use drugs that a general practitioner could handle. So one of the real limitations for the treatment of lymphoma, and actually, several other cancers as well, is so many of the tools in our toolbox on the medical oncology side require the use of injectable drugs that require special handling, right? So you need special equipment and special protocols and special waste disposal and all these other things in order to be able to handle these drugs in a safe way. And that's something that certainly can be done in a general practice situation, but it's really expensive and it requires a lot of time. And unless a general practitioner is doing this like every day, it's usually more of a commitment than is practical for them to make. So there's an awful lot of owners out there, I think, where one of the main sort of stumbling blocks or limitations to their being able to get care for their pet with lymphoma is I'm 5 hours away from the closest oncologist who is equipped to handle injectable chemotherapy drugs. Wouldn't it be nice if there was something that we could offer that, yeah, maybe isn't as good as the gold standard, but it might be better than palliative care? So, that's one of the studies that we're doing right now, is actually looking at this new protocol. We're calling it the Apple protocol, and I can thank one of our research interns for coming up with that acronym. It's great. That yeah uses two oral chemotherapy drugs and one injectable chemotherapy drug, or one injectable drug that's not cytotoxic chemotherapy. So despite the fact that it's an injectable, it can be handled in a routine clinic situation. And we have enrolled 30 dogs onto this treatment protocol. And all of the dogs have been enrolled. Probably 10 of them or so are still cooking. So we need a little bit more follow-up on those patients to really know, do we think that we have something here that's going to be useful as an alternative for those owners who can't travel to see a specialist. So yeah, that's one that I'm really excited about, just because I think it has the potential to impact a lot of dogs that, again, as you mentioned previously, there are places in the country where there are no, medical oncologists, right? So is this something that they could actually have done locally and provide some amount of benefit? Once again, maybe not as good as the best thing that's out there, but maybe better than doing nothing. So we're pretty excited about that. A second one that I'm personally kind of excited about has to do with this concept of targeted therapy. So I'm sure this is a concept that has been mentioned on your show in the past. This idea that we can do some fancy molecular tests on a dog tumor, and then pick some drugs that might work against that particular tumor. So it's a great, great, great concept in theory, but there are many, many practical things that limit our ability to, really successfully do that. Again, part of it is a lack of understanding about the mutations that are in dog tumors, or the other molecular alterations that are in dog tumors. But another huge, huge, huge limitation is we don't know how to give the drugs, right? So we're taking human drugs off the shelf, and usually a compounding pharmacy is, sourcing these drugs from who knows where and stuffing them into capsules and then selling them. And we don't know how much to give. We don't know how frequently to give it. We don't know if the amounts that we're giving are enough to actually do any good or not. So we're kind of taking it on faith at a number of different levels that these drugs that we're giving are going to be effective, or have a chance of being effective even. So, like I said, I mean, there's a whole lot of different reasons why this concept is maybe not quite ready for prime time in dogs, but we're trying to sort of chip away at one of those, which is if we sort of use the accepted dose for these medications, like the dose that you'll find that people are using out there, we're actually looking in the blood of dogs who get these medications, even a single dose of these medications, and asking, all right, how much of these drugs is actually showing up in the blood? And are we in the ballpark range of having enough drug show up in the blood to do any good? If we give the amount of drug that people are using out there. So I think that's an incredibly important piece of the puzzle. Again, there's lots of other pieces to this puzzle, but at least we can answer the question, all right, if you get one of these drugs from a compounding pharmacy and give the recommended amount to your dog, do you have any prayer of that tumor seeing enough of that drug for it to do its job? And I think that's just one little piece of very practical information that we can add to this whole targeted therapy thing that's sort of chipping away at one of the big knowledge gaps that we have. So kind of excited about that one too. And that's actually a study that's funded thanks to a very generous donation that we received here at CSU, that's earmarked very specifically for canine cancer. So this particular donor was very interested, again, specifically in funding research that is exclusively for the benefit of dogs. So not because it's going to look at things that are going to inform human cancers, or nothing that has to do with cats. So very specifically canine cancer. And that's a recurring source of funds that we'll potentially have at our disposal in perpetuity, which is really, really exciting for us, kind of being able to push the field forward.
That's wonderful. Yeah, we need more like that. We need more donors like that all around. So
Absolutely.
is that targeted therapy for different tumor types, or are you focusing on a specific type of cancer right now? And then since it's a funding source in perpetuity, that you'll then once you work on one tumor type, then you'll then expand it from there, maybe create a platform, expand it from there.
This study is actually looking at 5 different drugs. And again, each dog is only getting a single dose of one of these drugs. So we're purely just looking at the blood levels of the drugs. There's no therapeutic intent whatsoever. We're not there yet, right? We got to answer question 1 before we can answer question 10, which is, does it work or not? And so this is all comers. So right now, doesn't matter what kind of cancer a dog has, if an owner is interested in participating, again, their dog gets just one little dose of this drug, and then we just measure how much is in the blood over 24 hours. So it's a very, very simple study designed to answer a very, very simple question. One of the hypothetical advantages of this kind of targeted therapy is the same drug could be useful for multiple different kinds of cancer, if you've got a good way to figure out which tumors it's going to work for. And there's other folks who are trying to figure that component out. We're working on the drug side right now, which is, can we give these drugs in a way that's going to be useful or not? And I think, the answer for some drugs is probably not. Maybe you can't get enough of this drug into a patient safely to do any good. But I think for a lot of the other drugs, maybe the answer is going to be, yeah, you sure can. But the only way to know is to ask the question.
Absolutely. Is it a large trial? Do you have a lot of dogs that have signed up for it?
No, I mean, for this study, we only need 5 dogs per drug. So, we look at one drug in 5 dogs, then we look at the blood levels, and then we move on to the next drug. So it's actually a very straightforward, very quick study to be able to do. So we're 2 drugs in, 3 drugs to go for this initial little set of stuff that we're doing.
Well, it's important work you're doing because targeted therapy is a big part of the future, I believe. And you're right. There's a lot of things that have to be figured out about it. But one of the other reasons why Craig recommended that I get you on my show, we talked last time he was on the podcast about the potential for a universal cancer vaccine. And I'm not sure he said you were working on it. I don't recall. He just said talk to Doug Thamm about that. Is that something you're working on as well?
Yeah, so we're actually participating in what we think is the largest clinical trial that's ever been done in cancer. That's actually looking at a preventative vaccine for potentially multiple forms of cancer. So, got to be very, very honest. I'm not the scientific brains behind this. So, I've been collaborating for more than a decade with a really scary smart guy named Stephen Johnston, who is a professor at Arizona State University in Tempe, Arizona, and also is the CEO of a company that's trying to develop this technology called Calviri, Calviri. And the idea of a preventative cancer vaccine has been kind of a holy grail for a long time, and it's been very, very challenging to do, with the exception of very specific forms of cancer. So I would argue that there are certain preventative cancer vaccines that are already out there. So on the human side, the human papillomavirus vaccine, the HPV vaccine, has, just reduced the incidence of cervical cancer by 90%. I mean, it's been an unbelievable success. But that particular vaccine, it's not directly a cancer vaccine, it's a vaccine against a virus. I would argue that what we've done in veterinary medicine with feline leukemia virus, so the testing and the husbandry changes and the vaccine have all really contributed to a dramatic reduction in these horrible, horrible, and universally fatal lymphomas that we used to see in young cats. So that's another great example of what, in effect, is a cancer vaccine, although those vaccines were actually directed against cancer-causing viruses. So that's very easy, right? So viruses look really, really, really foreign to the immune system, right? They look nothing like anything in the body. So it's pretty easy to convince the immune system to look at a virus as foreign and, go in and attack it when it encounters it. So that's pretty straightforward. Doing that with cancer is a lot harder. So yeah, there are little tiny things on cancer cells that are different than normal cells. but 99.99% of a cancer cell looks just like a normal cell. So it's way harder to actually trick the body into thinking that a cancer cell is foreign and tricking the immune system into trying to pick it off than it is with a virus. The other thing that's really, really tricky about a preventative cancer vaccine is most of the things that are abnormal on cancer cells aren't shared between the same tumors. So in other words, if you've got two patients that both have an osteosarcoma, the abnormalities in those cells aren't going to be the same. So it's very, very hard to find shared, what are called antigens. So shared, abnormal proteins that actually serve as good targets for vaccines. If you're only looking in one particular place. So historically, everybody's always looked in the DNA to find mutations. And some mutations in the DNA turn on cancer-promoting molecules. Some mutations in the DNA turn off cancer-suppressing molecules. And these are the things that, everybody knows tend to cause cancer to form. Problem is that most of those DNA mutations are different, even between individual tumors that look the same under the microscope. And that's one of the things that's been really, really challenging with being able to develop, a good preventative cancer vaccine is you don't know what kind of mutations that cancer is going to get. So how do you design a vaccine against it? So my colleague, Dr. Johnston, so his big three line, so it's not mine.
No, then it's mine. My apologies for that. Okay.
Okay. So, Dr. Johnston's sort of contribution to this field is that he looked in a new place to actually find a source of abnormal proteins. And rather than looking in the DNA, which is where everybody else has looked, he's looked in this important intermediary molecule called RNA. So for those of you who may not remember the biology that you learned in high school, DNA is the template. It sort of tells the cells what to make. Proteins are the results. But in between those two, there's a messenger called RNA. And it turns out that there are changes in the way RNA is made that actually result in a whole bunch of altered proteins that are actually the same across different individual cancers, and even the same across different types of cancer. And so these particular errors in RNA are what form the basis for the vaccine that's being tested now. So the study that we're currently doing, it's us at CSU, University of Wisconsin-Madison, and University of California, Davis have enrolled about 800 dogs. These are all normal, healthy, middle-aged to older dogs that are either at, because of their breed, either at average or higher than average risk of death from cancer. So these are mixed breed dogs, and then a whole bunch of purebred dogs that, again, statistically are at that increased risk. So all these dogs were intensively screened at the time that they initially started the study to make sure they were healthy. So they got blood exams and urine exams and chest x-rays and abdominal ultrasound, and every little lump and bump on their body, was poked with a needle and looked at under the microscope to make sure that all of these dogs were totally healthy when they started the study, didn't have any hidden cancer that nobody knew about. And then these 800-ish dogs were actually randomized 1-to-1 to either get the vaccine that I just mentioned that's made up of these funny RNA-derived abnormal proteins, or a phony vaccine that looks exactly the same, has the same ingredients, just all mixed up in a random order, effectively. And then these dogs have been followed for up to 5 years. And the simple question is, who gets more cancer? The dogs that got the real vaccine or the dogs that got the phony vaccine? And this is all blinded. So I like to say this is actually a triple blinded study. So in humans, they have double blinded studies. Ours is triple blinded. So we, the investigators, don't know who's getting what. The owners don't know who's getting what. And the dogs certainly don't know who's getting what either. So it's triple blinded. And yeah, some of these dogs have been followed for 5 years. The end of this month actually represents the 5-year anniversary of the study. And we're about to sort of do our big analysis for effectiveness. So all along the line, we have been doing safety checks to make sure that, hey, everybody's doing well. There aren't any weird side effects happening. Yep, we can say that it seems to be very, very safe. But yeah, very, very soon, within the next month or two, we should actually sort of have some information about whether there seems to be any difference between the two groups of dogs. And I think there's a few different ways that this could actually spin out that could be very interesting. So the grand slam, right, would be, a whole bunch of different kinds of cancer were all prevented in dogs who got this vaccine. So they never got it and, ended up just like living out their lives and having some other old age related problem. So that would be amazing if we were able to permanently prevent multiple forms of cancer. So hallelujah, that'd be great. But there are a lot of other endpoints that I think would also be very, very valuable if we don't see that. Maybe what we see is a delay in cancer development. So maybe sooner or later, the cancer cells are going to get smart enough to outsmart our vaccine. But like you said earlier, if it takes a couple years for that to happen, that's a couple years of really excellent quality of life, right? That those dogs are potentially having before they have issues related to a tumor that's developed. So that would be wonderful. Maybe not quite the same as permanent prevention, but still really great. Then the third option is maybe we're not going to prevent all cancers, but there's certain particular kinds of cancer where this vaccine works better than others, where we do see really substantial reductions in risk or delays or things like that. So maybe it works great for, I'm just making this up because I haven't seen the data, but maybe this works great for lymphoma. Maybe it works great for mammary tumors or whatever, but it doesn't work great for certain other kinds of cancers. And that, I would say, would still be really exciting. So, there's lots of different ways that this could spin out that could all be good. Of course, there's also the possibility that it's not going to work at all. That's why we do the research, right? If we knew it was going to work, we wouldn't have had to do the study. And I have to just give an enormous shout out to the financial sponsors of this study. So this, thanks to actually an extra year of funding that we received, is a $7.4 million study that was sponsored by a group called Open Philanthropy. So Open Philanthropy is a philanthropic organization that was actually founded by an early Facebook investor who has decided that they want to fund very high risk but high impact science. And actually, this particular study, to my knowledge, is the first study in the biomedical sciences that they funded. So most of the stuff that they funded has been engineering and tech and things like that. So we're just so incredibly grateful that this outfit actually decided to take a chance on this incredibly high risk intervention. And obviously, I think they're very, very passionate about wanting to improve the health for everybody. So, but I have to be very honest. I think if this was only something that was going to benefit dogs, they might have taken a pass. I think that they love that part of what's going to happen is maybe if this is successful, there's going to be, a new exciting way to prevent cancer in dogs. But I think the other thing that they're very interested in is seeing this really provide some interesting proof of concept that this is something that should be looked at in people too. So that's not what this podcast is about, but we're certainly very hopeful that if in fact this does end up looking like something that's useful, that it's going to provide very strong support for trying to figure out if this same sort of approach might be useful in people. The other thing that's really neat about this study is that we have 800 dogs here that we've been watching really, really, really closely over 5 years. So these dogs get checked twice a year by by a specialist. They get blood work once a year. We're banking all different kinds of samples in these dogs. So I think at the end of the day, we're actually going to be able to answer a lot of other questions that might actually not be directly related to whether the vaccine worked or not. Right. So we actually have a manuscript that we're putting together about the screening results that we did. So we took 915 dogs and we did all these tests on them one time. Right. So these are all middle-aged to older dogs at normal or increased risk for cancer. What do you learn if you do this battery of tests on a whole bunch of old dogs? that's, I think, going to be a very, very interesting, result right there that we're actually getting ready to submit for publication. My colleague Jenna Burton actually presented that at the American College of Veterinary Internal Medicine meeting last year. So those preliminary results are out there. And again, at the end of the day, I think we have the potential to learn a lot about lifestyle. So what's the impact of obesity on the development of cancer and other diseases? What's the impact of geography? So do we see any difference in the kinds of cancer that developed in Davis, California versus Madison, Wisconsin versus, Fort Collins, Colorado? We've got zip code information on all these dogs. So are there certain zip codes where we saw different kinds of cancers developed? And can we correlate that with any sort of environmental exposures? So, I mean, I think this is a database that we're going to have the potential to kind of mine for really interesting information for a really, really long period of time. Very similar to, I'm sure you're familiar with the Golden Retriever Lifetime Health Project. So, it's, I think, the kind of information that we're going to be able to glean from this database is a little different, So obviously we're not following these dogs from young adulthood till death. We're not following as many dogs. They're not from as diverse an area across the entire country, but I think some of the advantages are we're not just looking at golden retrievers. We're looking at a whole bunch of different breeds. And the kind of questions that we're asking are slightly different. So I think they're going to be very complementary data sets at the end of the day. I guess the final thing I'd add about this study is I think the other nice thing is that the individual dogs and their owners on the study actually benefit a lot. So they're being checked over by a board certified veterinary oncologist twice a year for 5 years. They're getting free blood work once a year. Yes, we are seeing a lot of tumors in these dogs, right? Just like what you'd expect, but they're being caught, a lot of them are being caught very early. And early detection, as you know, is one of the keys to successful management of these tumors. So, I mean, I think as a group, a lot of these dogs are doing great with their cancers because they're being caught so early. The other thing is, part of what was baked into this study is actually a thank you in the form of a financial incentive for those owners whose dogs do develop a tumor. So owners actually get a little bit of a credit here at the hospital that they can spend on diagnosis and staging and treatment of a tumor that does develop as a thank you for having their dog participate in the study. So again, there's multiple ways that I think the individual dogs who are participating have the potential to benefit as well. So it's kind of a win-win-win thing. So maybe the individual dogs benefit, dogs as a group might benefit if this thing actually works, and then maybe in the very long run, people could benefit from this as well.
Yeah, well, 800 dogs over 5 years. That's quite a lot of data, and it's a wonderful study. That's big. My foundation's funded a few clinical trials, but they've always been small because, as you know, the more dogs you add to a trial, the more expensive it certainly gets. But it's a wonderful size study. So, how soon can we expect the results? Are you planning on publishing them, even though you haven't reviewed the data yet? Do we have a timeline on that?
So we're sending the data over to the statistician next month. It's hopeful that he'll be able to get back to us pretty quickly within a month or two. So I'm really hopeful that we'll be able to have some information to share, towards the middle or maybe the end of the summer. So coming up pretty quickly. And, we may start with trying to do a little bit of a late breaking abstract somewhere at an upcoming cancer meeting. But yeah, I would really hope that there's a paper out there to follow very soon thereafter. So stay tuned. Maybe just in the next month or two, we're actually going to have some interesting data to share.
Well, we'd love to have you back on the show when we get that data. When you said you didn't have it yet, I was hoping for a sneak peek. I thought you'd give us a little teaser of what the results of.
No, I
I'm
mean, like I said, I'm
excited.
blinded. I'm
Yeah.
one of the people who's blinded. I don't know who's getting what. So we're going to keep it that way. So we have one more year that we're going to follow these dogs. Again, thanks to Open Philanthropy, actually giving us an extra $1 million. But that's kind of like gravy. That's really going to be, hey, we're going to follow these dogs, learn even more about the tumors they develop, collect another year's worth of data on all the other stuff, all the non-tumor related stuff. That's going to be very, very interesting. But yeah, so the primary sort of cut date for the main endpoints is this month. So we'll hopefully know a little bit more quite soon. But I got nothing to share with you today, unfortunately.
That's okay.
Got to stay blinded.
That's all right. We'll have you back on the show with an update. But yeah, cheers to open philanthropy because we as you know, what I find is the comparative oncology that really sells our benefit for being able to study canine cancer. So it's a good thing that spontaneously developing tumors in dogs are very similar to that in humans, and we can use that. And we now have the one cancer model, I think, that's being adopted more commonly as well. So that's a wonderful study. I look forward to learning more about it. Well, we're almost at the hour mark. You're a busy guy. I wanted to talk about a couple of things. Are you the president-elect of the Veterinary Cancer Society, or is that outdated?
No, I am. So I think I assume the mantle of control at the end of this year. So
Yeah,
right now, Jeff Bryan from University of Missouri is our president, and I'm going to be taking over from him. I think it's January 1st.
excellent. Cheers to that.
And then I'm going to
Congratulations.
serve a 2-year term.
2-year term. That's good to
Thanks.
hear. But you're losing Barbara, though. I've learned. I learned that recently. You're losing Barbara pretty soon.
I don't think I know who that is.
Oh, I thought it was Barbara that helps manage the BCS. I might have that confused with somebody else. That might be my bad. I thought.
Um, yeah, Barb McGee, he was our old director from about 10 years ago.
Okay, I guess maybe she's no
Yeah,
longer.
so
I guess maybe she's retiring.
she's sorely missed.
Yeah. Okay. Okay. Maybe I got that confused. All right. And you
Yeah,
also
you're thinking of Sandy Struther. So Sandy is
Sandy.
actually retiring at the end of the year.
Sandy, gosh darn it. That's my memory. I got the Alzheimer's gene. So yes, Sandy, my apologies to Sandy. I've met her many times. We know her by virtue of our foundation. We've been very supportive of the Veterinary Cancer Society. Ginger has always attended more of the conferences and events that I have by virtue of my travels. But yes, Sandy, I got her name wrong. Sorry about that. All right. Well, Dr. Thamm, is there anything else as we wrap up? We like to keep it at an hour. You're a busy guy now that you're the president-elect of VCS, and you're also, it seems like you're an editor of a journal. I thought we'd have time for that, but we'll get you back on because I'm excited to learn the results of the universal cancer vaccine, and we could talk about some more stuff then. But before we go, is there anything else, any other message you would like to leave for the pet parents in our audience? And also, we have some veterinarians that watch the show as well. Any last words?
I don't think so. Just to rehash the stuff we talked about at the very beginning, go in and talk to an oncologist if your pet's diagnosed with cancer. You might learn some interesting things. You might find some good treatment options. And if nothing else, I think you'll be glad you got the information. So don't hesitate to reach out to a specialist. Cancer treatment in dogs, we're getting there, but it's very, very different from cancer treatment in humans, and it's all about quality of life.
Absolutely. Well, it's a good thing we have researchers out there like you that are pushing the envelope to acquire more and more data, and also that you're preoccupied with the interesting questions that need to be answered. So thank you, Dr. Thamm, for the work that you do at the Flint Animal Cancer Center there at CSU. Thank you for what you do.
Great. Thanks so much. Glad to be here.
Yeah, thank you for joining us for this episode of Fuzzy Butts and Friends. That's it, everybody. But before we go, I want to give a shout out to our sponsor for this episode. It's Genie's Therapeutics. They've got a wonderful hemp-based oil CBD product that I use for my very own Fuzzy Butt number 4, Indiana. You can learn more about their wonderful CBD ointment at geniestherapeutics.com. That's it for this show, everybody. We'll see you next week. Maybe we'll have our co-host Ginger Morgan back. You never know with her. Until then, puppy up. Talk soon.